Hello and welcome back. This is part 2 of 3 of our look at the male reproductive system and a little bit about fetal development. This is Professor Young, and we are continuing with our story, this time specifically looking at hormone production and function. Where are all of these hormones coming from within the male reproductive system? We know that testosterone is produced and released. We know that testosterone can be cleaved and turned into, through enzyme action, into a number of other types of hormones, DHT, dihydrotestosterone, as one example, estradiol or estrogen as another example. But where exactly is all of this testosterone being produced from? And so that's one of the things that we need to look at through this WIMBA session. And then we're going to talk a little bit about the impact of testosterone, or the lack thereof, when it comes to fetal development. In other words, genetically we understand that, yes, a male is XY and a female is XX. But how does that genetic code lead into specifically the production of ovaries, the production of testes, the production of the oviducts, the production of the vas deferens, so on and so forth, as we look at both female and male parts. And so that's what we're going to be looking at here. And so why don't we go ahead and get started. We need to go back into our seminiferous tubules that we looked at during the last session. Remember that the testes are the site of the initiation of sperm production. Remember that the spermatogonium, or the germ cells, are not under the direct direction of testosterone. Remember, there are no androgen receptors within that layer, and so it is definitely estradiol that starts the production of sperm. But that estradiol has to be created from something, and we understand that the basis for the formation of estradiol is testosterone. In other words, testosterone is produced and then cleaved. Well, where in the world is this testosterone coming from? It can't be too far away, because of the isolation that the testes have from the rest of the body. There's a reason why testes are external genitalia for males. There's a reason for that. And so where within the testes is testosterone produced? We understand that one of those areas are the Sertoli cells, but that's not the end. And in fact, we have another player that is definitely producing the large majority of testosterone found within the testes. And they are the Leydig cells. Now, Leydig cells are found within the interstitial spaces. In other words, in between those seminiferous tubules, not in the lumen, but physically in between the seminiferous tubules, are these cells known as Leydig cells. And it's the Leydig cells that are specifically geared towards the production of testosterone. That is primarily their role. That is what they do. All right, now, everything that you're looking at here, and when I say here, I mean here. These are all examples of androgen-based hormones. These are all examples of hormones that are further refined or further transformed or further produced or cleaved through enzyme action from testosterone. Testosterone is the parent here. Keep in mind, Leydig cells are responsible for 95% of all testosterone production. 95% of all of the testosterone produced within males comes from the Leydig cells. Another 4.5 or almost 5% comes from the adrenal gland. Very small percentages come from within the Sertoli cells. About 5% comes from the Leydig, or from the adrenal gland, which is where, by the way, the females would also be producing testosterone. Females are producing testosterone from the adrenal gland, same place that the males are doing it. The reason why levels are higher within the males all comes down to the presence of those Leydig cells. Females lack them, males have them. And that's why the hormone levels are so much higher within males. Now, look at what is being produced. You have your dihydrotestosterone that we've talked about, DHT. Androstenedione is another derivative, again, of testosterone. DHEA, dihydroepiandrosterone. I'm sorry, I'm tongue-tied. Is another one of those derivatives of testosterone. Now, DHEA can also be produced from the anterior pituitary as well. And so there's a little bit of dichotomy there as far as who's really regulating DHEA. Yes, DHEA can be produced from actually within the adrenal gland as well, but it's also can be cleaved from the testosterone. Keep in mind, all of those other guys underneath of testosterone are all responsible for the production of secondary sex characteristics and are a little bit more potent than what the testosterone themselves are. Now, as should be common practice by now, or at least common expectation, everything within the body is controlled by something else, which is controlled by something else, which is controlled by something else, which is controlled by something else. And so the question is, what exactly are the Leydig cells controlled by? In other words, how do the Leydig cells themselves know that they need to produce and release testosterone? The answer to that question is found again within the endocrine system. Remember what I said last session. Really, when we're talking about reproductive system, the bulk of the reproductive system is under the control of hormones, which means we're really just dealing with an extension of the endocrine system here. Take it back to the endocrine system. This is going back to week 1. This is like 15 weeks ago now almost. All right, the hypothalamus we know is going to produce a hormone known as gonadotropin-releasing hormone. You know that. If you don't know that, you forgot it, but you did know it. Gonadotropin-releasing hormone is released from the hypothalamus and acts on the anterior pituitary. It is the anterior pituitary that we are dealing with here. All right? And gonadotropin-releasing hormone specifically initiates the release of follicle-stimulating hormone and luteinizing hormone. Now, that may sound odd. If you know anything about hormones and if you know anything about female hormone studies, we understand that FSH and LH are primary female hormones because they directly relate to the development and maturation of eggs. Well, guess what? When you're talking about sex cells and you're talking about gametes, the egg and the sperm pretty much follow the same process of development. And so it would make sense that they would be under the same hormonal direction. And that's exactly what we see. Follicle-stimulating hormone are production of sperm. Luteinizing hormone is going to allow for sperm to mature. It's going to allow for the further development of the testes. And that's key. Gonadotropin-releasing hormone is released from the hypothalamus. It acts on the anterior pituitary to release the follicle-stimulating hormone and luteinizing hormone to the testes and specifically act on either the testes or more directly the Leydig cells. Does that make sense? Testosterone that is released from the Leydig cells have the ability to diffuse across the seminiferous tubules and enter into the Sertoli cells where, if you remember from the last session, the Sertoli cells can take that testosterone and cleave it into DHT, which is a more potent androgen, and use that DHT to further mature the sperm. Furthermore, follicle-stimulating hormone is going to stimulate mitosis. Now, where is that mitosis going to be happening? Yes, I expect you to scream back at the computer screen. Yeah, it's happening within the germ cells. And so follicle-stimulating hormone, when introduced into the testes, can also go ahead and spur on sperm production. Doesn't that make sense? If FSH and luteinizing hormone can act in the maturity of sperm, then why can't it also induce sperm production? If it's maturing the sperm to get rid of it, it also needs to be able to direct the replacement of those sperm. And that's exactly what we see happening. Right? Here's the other thing it does. Follicle-stimulating hormone is also going to be involved in production of testicular estrogen. What do I mean by that? What I mean by that is the estrogen that's produced from cleaving testosterone in the testes. So it kind of stays all within the same nice, neat little package. Males typically do not have free roaming blood estrogen. Estrogen that is produced within males stay and act localized. We see it in the testes. We'll see it again in the brain as well. In other words, testosterone will travel to these different areas, and then once upon reaching those levels or those regions, will then transform itself into the estrogen. It'll cleave itself into the estrogen. Now, here's the most current research as far as where each of these hormones are acting. So in other words, when we look at the germ cells and the initiation of spermatogonia or spermatogenesis, the very early immature fetal sperm, if you would, we know follicle-stimulating hormone is playing a role. These sperm have to mature. We understand that testosterone is playing a role because we know that testosterone is being converted into DHT, and that DHT is acting on the Sertoli cells to also aid in maturation. So that makes sense. As the process progresses into a primary spermatocyte and a secondary spermatocyte, and we don't think that there's really too much more happening as far as hormones go here in this area here. But look at the progression here, though. we understand, or we at least know, that testosterone does continue to play a role in spermatocytes maturing. So in other words, to go from really infantile sperm, spermatogonia, and as the spermatogonia develop, we don't think that there's any other real hormones that are involved outside of follicle-stimulating hormone and testosterone. And we also don't think that beyond that initial injection of those, that there's any more hormones that are really needed to have this happen. It's not until we get into those primary and secondary spermatocytes where we see testosterone. But follow this up. Check this out. Come down to where it says spermatids and spermatozoa and spermatogonia. Spermatids and spermatozoa. Spermatozoa is what's leaving the testes and entering into the epididymis. We see follicle-stimulating hormone and luteinizing hormone being reintroduced back into the picture. Why? Well, spermatogonia is the production of more sperm. And so if we follow back, we know that luteinizing hormone is going to act on the Leydig cells and allow for sperm to mature. Let's get out the old batch. We know that follicle-stimulating hormone is going to cause the release of testosterone, which is going to then be cleaved into DHT to allow for the new guys coming in to be maturing. But we also understand that follicle-stimulating hormone is also going to act on the germ cells to cause mitosis. And so what would be even more appropriate up here would be an arrow that is going back to the beginning. These guys, the spermatozoans, exit stage left. These guys are the ones that are heading out to the epididymis. And it's the spermatogonia that's now coming in to replace the spermatozoa that are moving on. Moving on down to the east side. All right. And so this diagram here just kind of reinforces what we talked about on the previous side. When we talk about hormones that are at play with the development of, there's nothing else we can add to this as of right now. As our understanding increases, as new research comes out, may things change? Sure. Once you go on for your master's or once you go on for your doctorates, those of you that are going to be doing that, maybe that research will be there. As of right now, this is our understanding. This is where we are. And so let's go ahead and talk a little bit about fetal development, because really testosterone is the main driving force when we talk about whether or not a fetus is going to become a male or a female, regardless of the genetic component. And what I mean by that is it's very possible to have a male XY genetically because of the low testosterone, because of missing enzymes that cannot convert testosterone into DHT, because of missing receptors. You could have a genetic male that develops as a physical female. Let me go over those reasons again with you in case you missed that. You can have a genetic male, XY. But if the male is not producing enough testosterone, or if the male is missing the enzyme to convert testosterone into DHT, dihydrotestosterone, or if the male is missing receptors to bind DHT or testosterone, you can have a genetic XY male develop as a physical female. And all of that relates back to the developing fetus. All of that dates back to the first trimester Weeks 7 and 8 are critical when we talk about going from a sexually indifferent fetus to actually developing sexual structures that will identify that XY male as being a male or that XX female as being a female. When we look at this, it comes down to, is the testosterone there and can it bind, or is it not there at all? It's testosterone that drives the changeover from being sexually indifferent to sexually identifiable. And that's what we need to look at for a few minutes. When we have a genetic male, XY male, on the Y chromosome, and by the way, this is why it comes down to the presence of testosterone or not, because we're dealing with the Y chromosome. That's what we're dealing with. Females don't have a Y chromosome. No Y chromosome, no high levels of testosterone, we develop as a female. Y chromosome, lots of testosterone, you develop as a male. And there's a gene that has been identified on the Y chromosome to affect the differentiation between being sexually indifferent and sexually identifiable. And it's called the SRY gene. The sorry gene, as we call it in the field. The sorry gene. That's why guys always say we're sorry. Genetically, we are programmed to do that. It's in our Y chromosome. Stop yelling at us. It's not our fault. Well, maybe it is, but this is a really good genetic excuse that we can try. The sorry gene is the sex-determining region of the Y chromosome. It is the area that will go ahead and basically code for an enzyme that will develop and produce and mature the testes. In fact, we call it testes or testes determining factor, TDF. If the sorry gene codes for TDF, the embryo will develop testes and will develop as a male. If the sorry gene is not present, TDF will not be produced and the developing fetus will develop as a female. That's the case when you have an XX. Now, there are some other things that come into play here as well, because testes determining factor also then controls, at least in the fetus, testosterone production. Remember, this is happening very early on. This is happening week 7. This is still the first trimester. This is still very early on within the pregnancy. Prior to the stimulation by the sorry gene and TDF, the fetus is sexually indifferent. It contains the basis of both the male and the female. In other words, it's got embryonic tissue that can develop into the ovaries and the oviduct and the cervix, and the uterus, and it's got tissue side by side that can develop into the testes and the vas deferens. They're both there. For the females, we refer to that as the Wolffian ducts. For the males, we refer to that as the Mullerian duct. If testes determining factor is present, it will initiate the production of the Leydig cells. Leydig cells will release testosterone. Testosterone will be cleaved into DHT. The testes determining factor and the DHT will both work on the embryonic tissue that is designated to become the testes and the vas deferens. If, and there's a little bit of a delay in this, but if TDF is not present, Sertoli cells will not be produced, testosterone will not be released, and therefore ovaries will develop. Now, remember what I said. There's a little bit of a delay here. Notice that male differentiation happens during week 7 and 8. Ovary production, ovary development doesn't occur until week 9. We have about a week to a 2-week delay between the switchover between a male and that of a female. And that makes sense. What if the testosterone needs a little bit more time to get up to speed, to get the levels up high enough? What if TDF needs a little bit more time to work? What if you have a couple-day delay in this process? If you have a delay in the release of TDF, and if you have a delay in the development of Sertoli cells, and if you have a delay in the development of testosterone converted into DHT, and you don't account for that, then you're going to have a higher instance of XY males that are developing as females. Testicular feminization syndrome. In other words, you have a genetic male that develops as a female. Now, there's a lot of ways in which that can happen, and we talked about that in lecture. But it's critical that you allow for a little bit of lead time. So that way you don't have ductwork being reabsorbed that shouldn't be reabsorbed, or you don't have ductwork that shouldn't be developing that is now developing. And so that's why we see that lag time of about a week to 2 weeks between the development of the male structures, embryonic tissue, and the development of the female embryonic tissue. What does it look like? What does it look like? Well, here's... I found a mistake in my PowerPoint. No one caught that. These guys should be reversed. I apologize. That should not be female. And that should not be male. I'm going to make that known right now. This is male. This is female. All right, let's make that new. When you look at this diagram right here, when you look at this diagram, you can see this is what the embryonic tissue basically represents. This is in and around week 6. This is exactly what the developing fetus looks like. Week 7, we begin to see the transformation in the development and the commitment to being physically male or female. Again, you can see right here. Hold on a second. This here would be the early precursor to an ovary. And then this here would be the further development of the oviducts or the fallopian tubes. Right here. So that would be the Mullerian system, that of a female. And if you switch to the other side, you can see right here, this would be the development of the male, right there. Right? And this here would be basically the immature testes. If the SARI gene is there and it's working properly, it's going to release TDF, which is going to create the Sertoli cells, which is going to help to activate the production of testosterone, which is going to cleave it into DHT, which is going to allow for the maturity of the Wolffian system. How does the body know to get rid of the embryonic female tissue, the Mullerian system? Glad you asked. Right, there is something that is referred to as MIS. Which stands for Mullerian inhibiting substance. In the presence of TDF and Leydig cells and testosterone, DHT, MIS will also be produced. MIS, Mullerian inhibiting substance, is a protein that will direct the reabsorption, the shrinkage, and the deteriorating, if you would, of the Mullerian system. If the SARI gene is not present, that means TDF is not present, which means which means Leydig cells will not be developed, which means testosterone will not be released in high quantities, which means it can't be converted over to DHT. Then MIS would not be released, and that will then lead to the maturing of the Mullerian system with the reduction, breakdown, and reabsorption of the Wolffian system. That's what all of this means. Now, again, if you need to back up, re-listen to that, and come back, do that now. You can pause this and rewind this a little bit if you so need to, right, which I would rather you do than to not do. This here is just a pretty little picture that kind of shows what happens if you don't have testis determining factor versus if you do have testis determining factor. And so on the left side of your screen, over here, you basically have the formation of the male. And then on the right side, you basically have no testis determining factor. How exactly are the Mullerian system going to develop? And so you can see that right there. That will not be on the exam, but it's just to help clarify exactly where we are going here and what we're kind of talking about. All right. Again, this is nothing that we haven't already discussed. All right. We know that in the presence of TDF, Leydig cells will be produced, which is going to produce testosterone, which is going to be cleaved into DHT, and that a combination of the TDF with the DHT and the testosterone is going to allow the Wolffian duct system, that the embryonic male tissue, to develop. And it will develop into the epididymis, it will develop into the vas deferens, the seminal vesicles, the ejaculatory duct. It's going to help promote the development of the prostate. It's going to help promote the production of the Cowper's gland, and so on and so forth. The testes were already there. We don't need to develop the testes. That's already there. And it's not until, by the way, the second trimester where we have really the development of the penis, and late second trimester, early trimester, the descending of the testes down into the scrotum. One of the things that I should also mention here is that and I'll type this out for you, right the right testis directs the right sorry, I can't spell. The right Wolffian embryonic tissue, which means then that the left testis is going to develop or coordinate the left Wolffian embryonic tissue. This can also lead to cases of hermaphrodites, because if you have an issue with receptors or a lack of testosterone or a lack of DHT, so if you're missing an enzyme on one half of the body and not the other half, you can see where maybe the right side is going to develop fully into the testes in the male anatomy, but the left side may not. And so then you've got a true hermaphrodite where you've got both male and female reproductive structures that are legitimately present. Now, again, female embryonic tissue, the Müllerian system, is going to develop if there is no SARI gene, which means no TDF, which means no Sertoli cells, which means no testosterone, which means no DHT, which also means no MIS, no Müllerian inhibiting substance. And so in that case, ovaries develop, oviducts, uterus, cervix, so on and so forth. Right. So again, there's no real tricks there at all. Now, something we did not do in class, but I want to spend a little bit of time looking at it here, and then we only have really another slide or so left, and that'll be the end of this session, a little bit shorter of a session. But here's three riddles for which I want you to think about and see if you can answer. Right? Scenario one, if a male embryo has its testis removed, it will develop female sex accessory organs. Why? And so why would an embryo, if we go in embryologically and we snip, and we remove a testicle or remove the testes, that genetic male will develop as a female. Why is that? Case two. That same male from case one that's had its testes removed. If we give it testosterone, both Wolffian and Müllerian ducts develop. Why is that? Testes have been removed, but we give it testosterone as an embryo, and it develops as a true hermaphrodite. Can you imagine doing that in the research lab, by the way? Can you imagine having your job be to go into rat embryos and snip testes out and then give it testosterone, knowing that you've created a hermaphrodite? That's a side point. Your real question is, why does that happen? Now, scenario three. A normal male embryo, you haven't removed anything, nothing's been altered, no nothing, is given an anti-androgen. In other words, it's given a substance that blocks the effect of the androgen, the DHT, or the testosterone. And neither male or female develops. Now you've created a true sexually indifferent person. You've created someone that is neither male nor female. It is sexless. How does that happen? My suggestion is pause this right now. Stop it, pause it, don't go any further until you have an answer to those three scenarios. I'm not going to give you the answer. That would be way too easy. In lecture, I will give you the answer, but I'm not going to give it to you here. I want you to think about this. If you have questions, shoot me an email. If you want to email me your answers, that would be good too. But I'm not going to give them to you here. I want you to think about those three scenarios, and you tell me what you think. All right. Now that hopefully you've paused, you've come back, you've thought about this, you've gotten some legitimate answers, hopefully. Now let's go ahead and continue on to the last slide. And again, this is nothing new. All right, DHT during the 8th week, which is, of course, is going to be produced by an enzyme that's going to cleave testosterone. Testosterone is present because the Leydig cells have been activated. The Leydig cells were activated by the presence of testis determining factor, which is present because of the SARI gene on the Y chromosome. All right, so in the presence of DHT, scrotum is going to form, penis is going to form. If testosterone is not present, which means DHT is not present, then penis doesn't form, but a reduced form of that is formed within the female as the clitoris, and the scrotum does not form. It remains separated, and it forms the labia minor and the labia major. Keep in mind, embryonic tissue can serve dual purposes. The direction of the development is going to be based on the presence or lack thereof testosterone and or DHT. So the clitoris is the same exact tissue that forms the penis, and the labia minor and the labia major is the exact same tissue that forms the scrotum. Just one testosterone is present, the other testosterone is not present. And so on that note, I will leave you scratching your head, thinking ever so diligently about those three case study questions that I gave you on the previous slide. You are going to shoot me an email if you have any questions about that, or if you just want to see if you're right or wrong. And I will leave you to review and ponder this stuff. The third installment of our male reproductive system is going to have us look at a little bit about the masculinization of the brain or the feminization of the brain. And then we're going to get into some of the things that happen when everything goes haywire. In other words, pseudohermaphrodites and hermaphrodites. And so that's what the third session is going to be about. As always, happy studying. And if you ever have a question, ever, please make sure you go ahead and shoot me that email and let me know, or ask me in class. And so until we come together again, have a good day, have a good night, happy studying, and we'll see you soon.